Questions? So remember that you need a broth culture of your unknown tomorrow. If it's been sitting in the incubator and it is growing, I would throw it in the refrigerator and then pull it out tomorrow for lab. And we're going to be inoculating a fair amount of media tomorrow. Consequently, what we're going to do is, in terms of inoculating, is instead of having you do pairs, we're going to have a side of the table work as a group. And so your side of the table will decide kind of how to split up inoculating the various known and unknown organisms. Because there's quite a few knowns that are going to be inoculated, and there's several media to do, so working in pairs is just too much. Okay? So I will let you go ahead and kind of organize as you see fit. As long as it all gets inoculated, that's fine. Friday, there will not be a new lab. Friday, we're just looking at the results of what we do on Wednesday. Okay? Okay, so we are talking about chemo-organo-heterotrophy. What does that terminology tell you about the metabolism of microorganisms? What strategy do they use? Huh? Okay, so a carbon source and an energy source are necessary, and in both cases they are using organic molecules. This is the strategy we use, obviously, in our metabolism, and this is the strategy that most microorganisms use as well, although other species do have other options. So within that particular umbrella, we've been talking about 3 different types of metabolic pathways, and they are aerobic respiration, anaerobic respiration, and fermentation. What do the first 2 have in common that the third one does not share? An electron transport system. Okay? What's the difference between the aerobic and anaerobic? What's your terminal electron acceptor? If it's oxygen, you're aerobic. If it is some other molecule, most typically an inorganic molecule containing oxygen, then it is anaerobic. What are you called if you can do both aerobic and anaerobic depending upon the conditions? You are facultative. Okay? You're a facultative anaerobic. Okay? So fermentation does not have an electron transport chain. There has to be some mechanism, however, to deal with the buildup of reduced coenzymes produced during glycolysis, because fermentation does have glycolysis. And so the pyruvate that's produced is going to have to be reduced into some final compound of fermentation using those electrons that are present on the reduced coenzymes. And that effectively recycles them. Different species produce different metabolic byproducts from fermentation. Different species have the ability to ferment different molecules. We talked about the fact, for example, that if you don't have beta-galactosidase, then you're not going to be able to ferment lactose, because if you can't break the lactose down into its 2 monosaccharides, then you can't ferment those monosaccharides. Okay? This lists some of the possible different kind of products, and the next slide does the same thing. I'm only going to show you this one. And you'll see that there are a variety of different substances that can be produced by fermentation depending upon the genes you have, because that governs what enzymes you produce, and therefore governs what pyruvate can be converted into. So there can be acidic compounds, there can be alcoholic compounds, there can be this compound you probably never have heard of before, 2,3-butadiene. I want to point out just a couple of things that are going to be relevant to lab tests that you were going to do. And this particular reaction right here, the reduction of pyruvate into 2,3-butadiene. This is a capability that can be detected by a test called the VP test. VP test stands for Voges-Proskauer. And so that particular test involves incubating your microorganism with some fermentable sugar, and then seeing if the organism has the capability to take the pyruvate that's produced into this particular end compound. Not everybody has the capability to do that. If you can do that, that is considered to be a positive Vogts-Proskauer test, and that's manifested by the chemical reactions that take place in the presence of the 2,3-butadiene turn your media red. And so red indicates a positive Vogts-Proskauer test. This is going to be a really important test for some of you, because if you have a gram-negative short rod, then there's a very good possibility that it is in that family Enterobacteriaceae. The enteric family. Things like E. coli and Proteus and Enterobacter. And they're all very closely related. So sometimes separating things into one genus versus another really comes down to one critical test. And this Vogts-Proskauer test is going to be in the key that you're using, one of those very critical tests. So you're going to come back and see this particular test later on. As you might expect, acidic compounds will lower the pH of the media in which the environment is growing. What's a convenient way we can determine whether the pH of an environment is lowering or increasing? What's an obvious thing we can do to see if the pH of an environment has changed as a result of fermentation? How do we measure pH change?
Look at the H plus or OH minus.
Okay, and to do that we can use a pH meter, for example, right? Are there other ways to measure changes in hydrogen ion concentration or pH besides the pH meter? You're familiar with the concept of a pH indicator? Okay. In general chemistry, did you use litmus? Okay. Litmus changes colors depending upon the pH. There are a number of pH indicators that are of a nature that at one pH they're one color and at another pH they're a different color. When we do fermentation tests, one of the pH indicators that's commonly found in the media is a compound called phenol red. And if the pH is greater than, I'm losing my numbers here, 6.9 I believe. I'll have to check that. Then the color of the phenol red is reddish color. If the pH drops below 6.9 and becomes more acidic, then the color of the phenol red turns to yellow. So by incorporating this pH indicator into a broth with some fermentable sugar and then incubating it, we can tell whether acidic byproducts are produced very easily visually just by looking at the color of the tube. So yellow tends to indicate acid fermentation. Okay. Now, some species of microorganisms produce just a single acid from their fermentation. So for example, I've used the example of souring milk, for example. The bacteria that are primarily responsible for causing milk to sour ferment the milk sugar, lactose, and produce lactic acid as a result of that fermentation. So they have the proper enzymes to produce lactic acid, okay? And that's all they produce. And that lowers the pH, and that causes the milk protein to denature, and therefore the milk to curdle. However, there are some bacteria, like E. coli, for example, this one's cut off here a little bit, that produce several different acids simultaneously. They can produce lactic acid, they can produce acetic acid, they can produce formic acid. And if you produce more than one type of acid, that is referred to as mixed acid fermentation. And mixed acid fermentation gives you a very, very dramatic pH decrease. Much so more than just producing a single acid. So there are differences in microorganisms in terms of how acidic they make the environment if they do a deep ferment. Some make it a little bit acidic, some make it really acidic. And the mixed acid fermenters do that. There is a test we will also do in lab called the methyl red test. Methyl red is also a pH indicator, but it changes color at a different pH than phenol red does. This doesn't change color until the pH drops really, really low. And so we're also going to do this methyl red test to determine whether your microorganism is a mixed acid fermenter as opposed to a single acid fermenter. So some of these things you are going to come back and encounter. It's really hard to discount the importance that fermentation has in a number of arenas. Okay? Some of these particular compounds are used by the chemical industry, and they are produced as a result of bacterial fermentation. Some of these compounds that are produced have a very dramatic effect on the nature of food if bacteria are fermenting that food. So the taste of kimchi, if you like the Korean equivalent of sauerkraut, or the taste of German sauerkraut, is as a result of the products that are produced during fermentation as those bacteria use the food resources in the cabbage that's being fermented, for example. The taste of certain types of cheeses is the result of the fermentation byproducts that are produced when the bacteria are fermenting the milk in the cheese-making process. So there's a lot of applications of fermentation to the food industry as well as the chemical industry. Sometimes the compounds that are produced are quite unique. So fermentation doesn't just produce acids and alcohols. Fermentation can produce vitamins. Fermentation can produce hormones. There are some cortisol hormones, some steroid hormones that fermentation can produce. And medically important, fermentation can also produce, in some species, antibiotics. So those important antibiotic molecules are oftentimes the byproduct of fermenting sugars. Now again, not all bacteria have the ability to make antibiotics because they don't have the right genes to encode the right enzymes to do those particular metabolic reactions. Sometimes the products of fermentation are harmful and are considered to be cellular toxins. That's why some organisms are pathogenic, cause disease, because their fermentated byproducts damage the cells that these particular bacteria infect. I got a little bit ahead of myself, but let's go back to this for just a second. Another metabolic strategy besides chemoheterotrophy is chemoautotrophy. And the type of chemoautotrophy that is the most interesting are the chemolithoautotrophs. So what does that particular set of terminology tell you about the metabolic strategy of the microorganism? What's its carbon source? Inorganic, probably CO2. What's its energy source? Inorganic, some inorganic molecule. These are the organisms that I talked about the other day that are not dependent directly or indirectly on sunlight energy. They can live by growing on entirely inorganic sources. So these are the rock eaters. How do they obtain their energy? Well, they oxidize inorganic molecules, and they do that by donating their electrons to an electron transport chain. The molecules that can be oxidized are varied. There are all kinds of different ones. They might include things like nitrate and sulfate, iron compounds, phosphorus compounds. These particular organisms so only produce a modest amount of ATP, not nearly the amount that's produced during either aerobic respiration or anaerobic respiration. How come? Well, these particular compounds, like nitrate, aren't carrying as many electrons that can be donated to the electron transport chain. If I've got a molecule like reduced NAD, for example, donating to the electron transport chain, that's got a lot of electrons loaded onto it, which can be then offloaded to the electron transport chain. Inorganic molecules don't have as many donatable electrons, and therefore we're not going to get as much hydrogen ion gradient formed, and we're not going to get as much ATP formed. So the energy yields from this are really pretty minimal. One class of organisms that fall into this category of being these chemoautotrophic organisms are the methanogens. We already mentioned methanogens when we were doing our survey. And these are members of the RK bacteria. In methanogenesis, what happens is hydrogen is your source of energy that powers the electron transport chain. So they're feeding electrons into the electron transport chain, generating a hydrogen ion gradient. The terminal electron acceptor in the electron transport chain is carbon dioxide. And as a result of electron transport, those electrons are transferred from the hydrogen to carbon dioxide to produce a more reduced compound, methane. So these are the methane-producing microorganisms. Methane has a profound effect on the atmosphere because it is a very potent greenhouse gas. And so the evolution of methanogenesis really contributed to the warming of the atmosphere as a result of methane trapping heat and preventing it from re-radiating back out into outer space once it had been absorbed by the planet. Now, we need a balance of heat, obviously, and right now, methanogenesis is helping to push us over the threshold of what is a healthy amount of greenhouse gases. So now we're trapping too much heat. We're getting a little bit out of balance. The methanogenic RK bacteria are huge, particularly in the buried seafloor samples of life. It's thought that maybe as much as 1/3 of all life on the planet is found in these buried deposits underneath the sea of these methane-producing bacteria. And under pressure, the methane that's produced in these deep undersea samples underneath the sea floor, under pressure, the methane can be converted from a gas into a solid form called methane hydrate. And it looks like a chunk of snow when you dig it up. But what's different from a chunk of snow is that if you take this chunk of methane hydrate and light a match to it, it will burn. Okay? Well, what we're concerned about now with global warming is that as the atmosphere increases in temperature, oceans are warming up. And as oceans are warming up, more of this methane, which has been trapped in the solid form, is being converted back into the gaseous form, and the oceans are actually releasing methane from the stored methane reservoir. That's an interesting outcome of global climate change. So, we're going to get to photosynthesis in a second, to phototrophs as another strategy. But before we do that, let's talk about an application of what we've talked about so far. First day of class, we talked about the importance of microorganisms. And one of the things I told you was that life on Earth could not exist without microorganisms for a number of reasons. One important reason is that microorganisms are responsible for cycling nutrients through our ecosystem and turning them from one form into another. Most organisms, particularly plants, require certain forms of compounds in order to be able to grow. So one form of compound they can use to support growth, another they can't. So there are these phenomena that occur in our environment called nutrient cycles. And there's a nitrogen cycle, and a carbon cycle, and a phosphorus cycle, and a sulfur cycle. Okay? The nitrogen cycle is really, really critical because nitrogen is an essential plant nutrient. And if plants can't grow, they are the photoautotrophs, they are the producers of our ecosystem. We can't survive without plants. We also need the oxygen that they're producing for our aerobic respiration. So the availability of nitrogen in the proper form is critical for life on Earth as we know it to exist. Plants require a particular type of nitrogen. They require ammonium. Now that's not the only form nitrogen can occur in the environment. But if plants have ammonium available, they can incorporate this metabolically into carbon skeletons and make things like amino acids and nucleotides. If plants don't have ammonium available, they won't grow. Okay? They can't make these critical compounds. So this is an essential nitrogen source for plants. So much so that ammonium is considered to be a limiting factor to plant growth. So what have we been forced to do in our agricultural system? Because a lot of plants, as they grow, pull the ammonia out of the soil. We've got to resupplement the soil with more ammonia, or something that can be converted into ammonia. We have to fertilize. Okay? So nitrogen moves throughout our worldwide ecosystem and is converted from one form into another, into another. What converts it? Microbial metabolism. Organisms feeding on these different sources of nitrogen and converting them into new compounds is what drives the global nitrogen cycle. So we're going to look at the nitrogen cycle. Oops, forgot something. Get there in a second. We're going to look at the nitrogen cycle in just a minute and talk about how microorganisms are responsible for making sure that this particular critical phenomena takes place. Before I do that, I forgot one thing. There are a couple of nitrogen-containing reactions that are really important that we haven't talked about yet. Here are some of the reactions involving nitrogen that you might encounter in microbial metabolism. One of the things that is a possibility is that some organic molecule or organic acid, like pyruvic acid, if you add nitrogen to it in the form of ammonia, can be converted into an amino acid. That particular phenomenon is called amination. So we can aminate organic acids and convert them into amino acids. That's really important in the metabolism of many microorganisms, including ourselves. We can produce some of our own amino acids metabolically if we have the right substrates available. What do we call the amino acids we can't produce metabolically? You have to get them in your diet. You ever heard of essential amino acids? Those are ones that have to be obtained in your diet. Some other organism has to be able to produce them, like a plant. Okay? Once you've got one amino acid, another possibility is that one amino acid can be converted into a different one. That particular phenomenon is called transamination. So here we have aspartic acid, that's a particular amino acid. And there is a chemical reaction where alpha-ketoglutarate and aspartic acid can interact, forming glutamic acid, a new amino acid, and oxaloacetic acid. Where does alpha-ketoglutarate come from? You recognize that compound? Alpha-ketoglutarate is one of the intermediate compounds in the Krebs cycle. Once you have amino acids, it is possible metabolically to remove the amino group from an amino acid and form an organic acid. When might you want to do that? Well, proteins can be used as sources of carbon and nitrogen for not only human metabolism, but microbial metabolism. So what happens when you eat a protein? The first thing you do is you hydrolyze it into its amino acids by digestive processes. Some of that happens in the stomach, most of it happens in the small intestine. So now you've got these amino acids. You can then transport those amino acids across the intestine, into the circulatory system. They can be then moved into individual cells. And if you have the proper enzymes, you can then deaminate those amino acids, convert them into organic acids like alpha-ketoglutarate. Those are things that are part of the Krebs cycle. So you're feeding those molecules into the Krebs cycle. And then you're using those as an energy source to make ATP. Okay? So deamination is a critical part of metabolism of proteins. And what do you produce as a byproduct of deamination? Ammonium. Okay? Ammonium is an essential plant nutrient, but in humans, ammonia is toxic. So as humans evolve the ability to metabolize protein, They also had to evolve systems to deal with the excess ammonia that was being produced. What systems are those? What kind of systems do we have to get rid of ammonia?
We use the liver and then we feed out.
Yeah, our excretory system, okay? We can convert ammonium into urea, for example, and then we excrete the urea, okay? Well, microorganisms can do this as well, at least some can. And we're going to do some tests later on in lab to see, can your microorganism deaminate, remove the amino group, from the amino acid phenylalanine? And that's a particular test we're going to do. And some microorganisms can do it, and some can't. How do you suppose we might detect phenylalanine deamination? Look over here. How might we detect whether that deamination has occurred? Pardon me, Sonny? pH again, because ammonium is going to do what to the pH? It's going to increase the pH. It's a base, okay? And so if we have pH indicators that can measure an increase in pH, then that can be a good indicator of the fact that ammonium is being produced as a byproduct. This particular reaction of deamination is going to be important in this nitrogen cycle that we're going to talk about next. Okay, so let's talk about the nitrogen cycle. There are several metabolic processes that occur and are essential parts of the nitrogen cycle. One of them is the phenomena of nitrogen fixation. Another process is the process of ammonification. This is really the result of deamination. A third phenomena is the process of denitrification. And the fourth process is the process of nitrification. These refer to metabolic processes. So these are types of metabolism. And there are some organisms that can do this. There are some organisms that can do this. Some that can do this. Some that can do this. And typically what happens is that when one organism does one set of reactions, that produces a product. And then that product is the substrate for another set of reactions. So this depends upon this. This depends upon this. This depends upon this. Okay? If any one of those processes doesn't occur, the cycle stops. And if the nitrogen cycle stops, life on Earth stops. So organisms that might seem very insignificant, and you might think, oh, the world could do without them, might be very significant because they are the denitrifying bacteria, for example. And if we wiped all of those out, then the nitrogen cycle would come to a grinding halt. Plants would not have the form of nitrogen they need to grow. Without plants, animals can't survive. So this really is a clear illustration of what's happening in terms of the relationship between the importance of microbial metabolism and some little critter breaking something down to get energy and carbon skeletons and producing a product. But it's more than just their survival. They're part of a bigger scheme, in this case, a nutrient cycling mechanism. Now, in every cycle, be it nitrogen, carbon, sulfur, phosphorus, there is something called a reservoir. And that's where the material tends to be most predominant. There tends to be the largest store of that particular material. Does anybody know in our environment where the primary reservoir is of nitrogen? Where do we find most of our nitrogen in our world? In the atmosphere. Okay? So for nitrogen, it is nitrogen gas. It's the atmosphere. 78% of the atmosphere is nitrogen gas. Nitrogen is very inert. It's very stable. And plants can't just pull nitrogen out of the atmosphere and incorporate that into amino acids and nucleotides. It doesn't work. They don't have the capability to do that. However, there are some bacteria that do have the capability to reduce nitrogen gas into ammonia. And that particular process is called nitrogen fixation. Some nitrogen fixation occurs in a symbiotic relationship. Bacteria and certain genus of plants called legumes. Where do the nitrogen fixing bacteria live in the plant? In the roots. Okay? If you pull up the plant of a legume, there are growths on the roots that look like tumors. They're called nodules. Well, what's so special about the bacteria that can fix nitrogen? They have an enzyme called nitrogenase that can do this particular conversion. This particular enzyme is very oxygen sensitive. One of the things that the nodules do is that they provide an environment where the nitrogenase can survive. So there are some oxygen excluding mechanisms built into that nodule. Some things that are going to tie up oxygen and make sure it doesn't denature or poison the nitrogenase enzyme. Particularly in the nodules, there's a high concentration of a protein called leg hemoglobin. Leg refers to the fact that this is found in legumes. And what does hemoglobin do? Hemoglobin binds oxygen, doesn't it? So this is an oxygen scavenging or binding compound that is found in the nodules and tends to soak up the oxygen that might get into that nodule, which would otherwise inactivate that nitrogenase enzyme. Now, not all nitrogen fixation is symbiotic. There are also non-symbiotic nitrogen fixers as well. We mentioned, for example, that members of the cyanobacteria can be nitrogen fixers. So they've got the nitrogenase enzyme as well. They don't have leg hemoglobin. they have some other mechanisms available to try to bind up that oxygen. One of the things that you'll see in the cyanobacteria, and I think you might have seen this in botany. I think in botany you might have looked at Anodina, which is a cyanobacteria. And they have these little swollen football-shaped structures within the filaments of cells that made up that algae. And this particular little swollen structure is called a heterocyst. This is where that nitrogen fixation is occurring. And so I'm not sure what they are, but there are some mechanisms associated with heterocysts that help to protect that nitrogenase enzyme. Once you've got the ammonium produced, and if it's released into the soil, then other plants can absorb that ammonium and use that as their nitrogen source to make amino acids, to make nucleotides, and to support their growth. If this didn't happen, we wouldn't have a nitrogen cycle. Okay? So this shows you the reservoir of nitrogen, the nitrogen gas. And this process of nitrogen fixation, the bacteria living in the nodules of the legumes are converting that normally inert nitrogen gas using their special enzyme nitrogenase into ammonium. Okay? That is the first step in the nitrogen cycle. Those particular bacterium are providing the ammonia that plants need to grow. Now, what happens to those plants? They grow, they will eventually die. Their dead bodies are returned to the soil. But if that's all that happened, is if they just laid there in the soil, the nitrogen that they took out of the atmosphere would now be tied up in organic molecules that are part of their dead bodies. So we need some way to then convert the nitrogen that is now part of amino acids and nucleotides back into other parts of the nitrogen cycle, because what's the goal of any cycle? The goal of any cycle is to get back where you started. So the nitrogen will be pulled out of the atmosphere, initially by nitrogen fixation. But if the cycle is going to be self-sustaining, then the nitrogen's got to be returned back to the atmosphere again in the form of nitrogen gas. So that's what some of these other steps are involved in, is those other conversions that take nitrogen and turn it from one form into another until it finally returns to its original location, the atmosphere. Well, second part is ammonification. What's happening here? Here, there are various bacteria that have those enzymes, those deaminase enzymes, that can take the proteins and the nucleotides and things of those dead plants and animals that have eaten the plants and deaminate the proteins and convert the nitrogen back into ammonium form again. Some of that ammonium could be just reabsorbed by other growing plant roots, and so it can be used over again. But there are other possible fates for ammonium that involve bacteria that we'll talk about in just a second. You have experienced ammonification if you have ever come across roadkill, for example, and you go to check it out, and you get this wafting smell of ammonia coming from the roadkill. What's that the result of? That's the result of those bacteria deaminating the proteins of that dead organism and converting it back into the nitrogen form of ammonia. Okay? Another important part of the nitrogen cycle is, well, what if that ammonia isn't taken up, all of it isn't taken up by other plant roots? What else can happen to it? Well, that ammonium can be metabolized into other forms of nitrogen, nitrite and nitrate. That particular process is called nitrification. What kind of organisms are we talking about here? These organisms are the chemolithoautotrophs. Sometimes we just cheat and call them lithotrophs. And what are they doing? They're taking that reduced ammonia and donating those electrons to electron transport chains. And in the process, they are producing more oxidized forms of nitrogen. This particular process of producing these more oxidized forms of nitrogen, including nitrate, is called nitrification. Those particular organisms are called the nitrifying bacteria. Well, think what we've accomplished so far. First, we converted N2 gas into ammonium, and now the ammonium has been converted into more oxidized forms of nitrogen, like nitrate or nitrite. Nitrate is actually fairly toxic to cells. You might have heard of situations before because sometimes fertilizer contains nitrate. They go, oh, wait a minute, why would fertilizer contain something toxic? Well, we'll talk about that in just a second. But if fertilizer runs off into water supplies like a well, and wells get contaminated with nitrate, for example, that's a real health problem. Okay? High levels of nitrate can be lethal to humans, for example, if you drink water contaminated with nitrate. The last step of the nitrogen cycle is the process of denitrification. And denitrification involves the conversion of nitrate back ultimately into nitrogen gas. So these nitrates are reduced into nitrogen gas. What metabolic process is driving this? Anaerobic respiration. So where are those nitrate ions participating? They're terminal electron acceptors in the electron transport chain. And nitrate can be reduced to nitrite. Different organisms can then use the nitrite and reduce it into another compound called nitric oxide. And the nitric oxide can be reduced by different organisms into nitrogen gas. So one organism produces a product and the other organism uses that, and eventually the nitrogen is converted back into nitrogen gas. This particular phenomena is something we will look at in lab. And we're going to look at the process of denitrification. And particularly, we're going to see, does your organism have a capability of reducing nitrate into some other form of nitrogen? So we're going to do a nitrate reduction test. And that's going to provide some important information about your microorganism. So we'll do just a little bit of quick review about this on Thursday. We'll do a little bit of photosynthesis, and we may have time to get to the next section, start the next section, which has to do with how do we kill microorganisms. So I will go ahead and post all that material on Moodle, so you're ready to go with that.